All right, as promised, this is part two of muscle physiology, and pick up where I left off in video one. To make sense of this slide, I'll remind you of something we talked about in class. I made a list on the board that was titled something like, ways to increase tension in a muscle. And number one was recruitment, just add more motor units. Number two was preferentially use larger motor units, which every muscle has a range of motor units that are small, medium, and large. And so if you activate the bigger ones, the muscle will be stronger. And number three was increase action potential frequency to those motor units, which will also increase contractile force. The fourth one, which we didn't talk about the first time I went through the list, is optimizing sarcomere length. And that's what this graph is showing you. And I think the benefit of looking at this is it gives you a more realistic view of how actin and myosin are actually interacting with each other. And all this is showing you is tension, which you could read as intensity on the y-axis, sarcomere length on the x, and what they're calling 100%, the sort of default length, is where myosin and actin filaments perfectly line up with each other. Right? These gray bars are supposed to be Z-discs here. Right? And that's nice because every myosin head can make contact with an actin filament and they're all capable of pulling, and so potential contractile force is 100%. All this is showing you is that if the sarcomere gets too long, this is an extreme, but even anywhere in between, myosin and actin effectively slide past each other. That's why this whole system is sometimes referred to as the sliding filament model of muscle contraction. If they slide too far, there's nothing to pull on. It's almost like a rope being pulled out of your hands. You can't apply force anymore. The same thing is true here. If you contract too far and the muscle gets too short, right, these myosin filaments will also slide too far and not have anything to pull on, and they'll decrease their contractile force too. So that's why muscles actually don't really get all that much shorter when they contract. An actual total length change of a couple inches is pretty normal even for a very large muscle, because if you went too much further out of that range, contractile force would go down dramatically. All right. Okay, next topic, muscle twitch. So the idea here is that muscles, a contraction of a muscle is coded based on frequency, meaning action potential frequency. And the more action potentials you send, the more acetylcholine you release, the more calcium enters the muscle fiber, the stronger the contraction will be. What this is showing you is sort of an idealized single pulse, single action potential. And we call the response that a muscle has to one action potential a twitch. Now, this only really happens in a lab setting because in living people, you never have just single, standalone, all by themselves action potentials because, remember, resting tension is never supposed to be zero. But if you did, so if you set this up in a lab with a muscle that was taken out of an animal or a fresh cadaver or something, you could make this happen by hooking an electrode to it. So this red arrow, when they say single stimulus, that's supposed to be an action potential, and it comes exactly right there. What you'll notice is, and this may seem picky, but it shows you important physiology, this tiny little sliver of time right there, only a few milliseconds, there's a space where tension, which is y-axis, doesn't go up right after the action potential comes. That period of time is called the latent period, and it's said to be excitation-contraction coupling, which really just means going from an action potential to the point of actually producing tension, which means myosin pulling on actin. So everything in between there, the action potential cascading down T-tubules, calcium being released, calcium binding troponin, actin and myosin engaging, all of that is happening during the latent period. Once calcium is released and actin and myosin can actually contact each other, myosin starts pulling, you get tension, and that's the period of contraction. And that jives with the definition of contraction that I put on the board in class, which is just increasing tension. As soon as calcium starts being pumped back into the SR, which will happen fairly quickly, tension starts going down. And again, decreasing tension was our definition of relaxation, so that works out pretty well. The only other real variable in this is how fast those changes happen. Some muscles, or muscle fibers more specifically, are fast twitch, which means they contract and relax very quickly. Other muscles are slower twitch, which means they contract and relax a bit more slowly. And there are benefits to both. Obviously, being fast is important in some cases, like eye movement, but it comes at the cost of having poor endurance. And so muscles that are faster twitch generally tend to wear out more quickly, and muscles that are slower twitch, like the soleus, which causes ankle extension during walking, tend to have a lot better endurance, and they don't really need to be as explosive. Right. Like I said, this is a lab setting. You would never see a single, all by itself, action potential in a living person. And so what happens when you send multiple action potentials in a row? Well, you increase frequency. And this graph is showing you that if frequency gets high enough, again, each red arrow is an action potential. This action potential caused an increase in tension. You contract, you start to relax. But if before tension comes back down to resting, you send another action potential, you're essentially stacking the twitches on top of each other, where the twitch causes a contraction, tension starts to come back down a little bit, but you boost it back up before there's a complete relaxation. And so the whole thing is additive. You're adding or stacking one twitch on top of another. And what you get at the end of a series of additions is a sum, so we call it summation. And this is based on frequency because the faster the action potentials come, the greater the effect is going to be. If I made these action potentials come even more frequently, there would be less time in between twitches to relax, and we would go up even faster. Okay, so this is basically explaining how increased frequency causes an increase in tension or contractile force. At some point, if the frequency gets high enough that the muscle is effectively maxed out, the muscle fiber couldn't possibly produce any more tension, then you get a plateau. And that plateau is called tetanus. So this graph is showing you fused tetanus, which means the frequency of action potentials has gotten so high that no matter how many more you send, there's nowhere else to go because the muscle just can't produce any more tension. Every last myosin-actin pair that could possibly be binding has done that, and there's no way to increase tension. And the last thing I should mention is, remember that twitches, I keep saying muscle, but really one twitch happens in a motor unit, right? Not really in an entire muscle, because action potentials come down individual motor neurons to individual motor units. And so you would have to talk about this on a motor unit basis, not really an entire muscle kind of basis. All right, to illustrate that idea of tetanus, this is showing you, what happens when things get taken to an extreme. Most people aren't capable of even voluntarily activating their skeletal muscles to even 50% of their maximum potential contractile force or intensity. And that's obviously for safety reasons, so that muscles and tendons don't get damaged, and your brain puts a lot of controls on what your muscles are allowed to do. And if somebody gets a tetanus infection, Clostridium tetani, the bacterium that causes the infection, got its name because it causes intense muscle spasms and shows you, what would happen if those limits weren't put in place. And effectively, the toxin released by Clostridium tetani inhibits an inhibitory neurotransmitter in the spinal cord, and you basically turn off the brain's ability to turn off muscle contraction. And since all the inhibitory neurotransmitters blocked, the excitatory neurotransmitters run wild. Every skeletal muscle in the body contracts really intensely. And since they're all contracting at the same time, the stronger muscles win. In the upper body that mostly leads to flexion, and the lower body that mostly leads to extension, and that gives you that very standardized pattern. Everybody that gets tetanus effectively looks like this. And the end result of that is really severe joint damage, usually spinal damage from overcontracting the spinal extensors much too intensely. All right, as we've been moving through this, I've been trying to, in the notes I started off zoomed in really far, and then we've been slowly zooming back out to look at the big picture, and this sort of pulls everything together. When we went through definitions in class, I tried to make a really emphatic point of saying that contraction and shortening of a muscle aren't always the same thing. And I usually say that contraction is a muscle trying to get shorter, but it doesn't mean that the muscle is always successful, or even that you even want it to be successful, like when a person's lowering something. And so these are just putting technical terms on ideas that you're already familiar with. And the two main terms, isotonic and isometric. Isometric is the easy one. Iso means stable. Metric, like meter, means distance. So stable distance means the muscle isn't changing length. And all that means is the muscle is producing exactly the same amount of tension, or really torque, that the load is exerting on that muscle. And so just imagine holding something in front of yourself and keeping it perfectly stable. That's an isometric contraction. Isotonic, stable tension. If the stable tension is greater than the resistance, meaning more than what you're trying to lift, the muscle wins and it gets shorter. And so a contraction where the muscle shortens is called concentric. If you have stable tension where the load has a greater tension, meaning the object is heavier than the force you're exerting, obviously the muscle loses and gets longer. A muscle lengthening is an eccentric contraction. And practically, this basically means lifting and lowering. Lifting things is a concentric action, and intentionally lowering things is an eccentric action. All right, at this point, pretty big change of pace. We're going to talk about muscle metabolism, ATP production, and how muscle contractions are actually fueled. And the basics of this are fairly straightforward. Muscles, just like every organ in the body, prefer aerobic respiration whenever possible. It's by far the most efficient way to do things. And so if oxygen is available and there's enough oxygen to make enough ATP to fuel the muscle contraction, you'll do that whenever possible. The potential ways that you can make ATP aerobically, for the most part, are either glucose or fat from the blood. And the overview of this that I gave in class, I did a very quick recap of glucose moving through glycolysis, making pyruvate, that going into acetyl-CoA, acetyl-CoA being the starting molecule of Krebs cycle, the bottom of that coming out to an electron transport chain that produces ATP at the end. That ATP is the currency for making muscles contract. It's what myosin uses to reset, right, every time it goes through a power stroke cycle. And this makes a lot of ATP. Somewhere in the ballpark of 32 or so is usually a estimated quote. And you can do that with glucose. You can also do that with fats. Fats can be converted to acetyl-CoA through beta oxidation. And generally, slow twitch type 1 muscle fibers like to use fat whenever possible because you have lots of it. There's plenty of adipose tissue to go around to fuel muscle contractions when the muscle is either at rest or contracting very lightly. But blood glucose is also a possibility. As long as there's oxygen present, because remember that electron transport chain needs to use O2 to finish the job and actually produce ATP at the end. As long as there's oxygen there, you'll keep doing this as much as possible. The backup plan, there are really a couple backup plans, but the primary one is if oxygen isn't plentiful enough to make the ATP that you need to completely fuel the muscle contraction, you can take some of that pyruvate and ferment it into lactate, lactic acid. That's fermentation, right? Which means a lot of your pyruvate is still going through this aerobic pathway to make ATP with oxygen. You're just putting an additional amount this way through fermentation to make lactate. They both require glycolysis. And so if you're trying to maximize ATP production, you're going to use as much glucose and fat as possible. You'll maximize the aerobic part of this with oxygen. And then if you still need more ATP after that, lactate and fermentation get stacked on top, almost like a release valve, where you put the extra pyruvate out this way. It's just a way to get rid of it being the pyruvate, so that you can keep going through glycolysis and make even more ATP. The problem with this is one of the byproducts of anaerobic glycolysis is acid, and muscles become acidic. That's where the fatigue and maybe eventually the soreness of really intense anaerobic muscle contraction comes from. And so the aerobic part of this, people can do for hours and hours on end very efficiently, and doesn't lead to a buildup of acidity. The anaerobic part is great for really high capacity ATP production in very short bursts. But most people can't be anaerobic for more than, minutes at most before getting incredibly fatigued, because you're basically, acid burning those muscles from the inside out. All right. Maximizing oxygen consumption. This is showing you, I know we haven't done cardiovascular stuff yet, that's AMP2, but it's trying to give you an idea of how oxygen is provided to muscles. And all this graph is showing you is partial pressure of oxygen on the x-axis, which partial pressure is just how gases are measured quantitatively. So you can conceptually think of that as the same thing as concentration. Right? And the x-axis is O2 saturation. Hemoglobin and myoglobin are proteins that bind and hold on to oxygen. Saturation just is a measure of how full those binding proteins are. And so if hemoglobin saturation is 80%, it just means that 80% of hemoglobin's binding sites are full of oxygen and the other 20% are empty. Hemoglobin is an oxygen binding protein that exists exclusively in the blood. Myoglobin lives exclusively in muscle tissue, both cardiac and skeletal muscle. And that means hemoglobin circulates and myoglobin stays put. And so your hemoglobin circulating, you have very, very high PO2 in the lungs, which mean, very simply, in the lungs, saturation goes up. That's just a technical way of saying that hemoglobin picks up oxygen in your lungs. That hemoglobin in your red blood cells will circulate out to systemic circulation. It'll hit muscles, and since muscles are using oxygen, O2 levels drop, so that in a muscle, O2 levels might be anywhere between close to 0 up to 40. The 40 range being rest, and the low end being high intensity contraction, and then obviously the space in between being more moderate contractions. And so hemoglobin hits the muscle, PO2 drops, which means this line takes a free fall, and saturation goes down. That's exactly what you want, because saturation going down just means hemoglobin is letting go of its oxygen. That's what it's supposed to do, because muscles need that oxygen to produce that aerobic ATP that they're using to contract. The problem you run into is if the contraction gets hard enough that PO2 in the muscle is getting close to 0, you're running short on oxygen. That's where myoglobin comes in, which is your backup. Myoglobin lives within muscle fibers and is holding on to oxygen essentially as just a backup, sort of last resort. You've used, or you're maxing out the ability of hemoglobin to deliver O2, which never stops, but you can completely use it up in the instantaneous sense. If hemoglobin is maxed out and PO2 gets low enough, eventually myoglobin saturation starts dropping too, which is just saying you're using your backup. Myoglobin's letting go of its oxygen. That's also being used to make more ATP aerobically than you would otherwise be able to. The difference is hemoglobin keeps circulating, and so it'll keep going back to the lungs and pick up more O2 and come back again. Myoglobin doesn't circulate, and so when myoglobin lets go of its oxygen, it's pretty much gone until you rest again. Because when the muscle stops contracting and you rest, right, PO2 starts climbing back up, and that oxygen that hemoglobin is bringing in is going to be used to resaturate myoglobin. The technical term for this is oxygen debt, which is essentially saying you borrowed some O2 from myoglobin while you were active, and now that you're resting again, you got to pay back the debt. And that's part of the reason why people will continue, breathing heavily and having an elevated heart rate even after they're done exercising, because they have to resaturate myoglobin. That'll eventually get hemoglobin levels back up to normal until you're in that resting, 35 to 40 range where muscles like to be when they're not contracting. Okay. All right. This slide is just showing you that the source of energy for aerobic respiration is variable. I'm not going to go into a lot of detail other than to say that muscles, especially when they're working aerobically, can use either glucose or fats, and either one works as far as producing ATP. There are benefits either way. My point is, muscles kind of have to pick one to really, really specialize in. Most people are fairly carbohydrate focused because typical diets have a lot of carbohydrate in them. And so if you take people that are eating a lot of carbohydrates, this experiment measured their ability to burn fat, to oxidize fat during exercise. And if you take people that mostly eat a high carbohydrate diet, they're not very good at burning fat. And if you take people that have been put on a ketogenic diet, which means they've been forced to eat lots and lots of fat but very few carbohydrate, obviously they become really good at oxidizing fat. It doesn't really dramatically affect performance either way. The muscle still contracts, but the mitochondria in those muscles can adapt to become specialized at being very good at going through glycolysis and using glucose, or very good at going through beta oxidation and using fats. And either is possible, but it takes time to adapt from one to the other. All right. The anaerobic part of this I've already pretty much talked about, but all I'll say in summary is anaerobic respiration is always a last resort. You never really use it unless it's absolutely necessary, because you can't make the ATP that you need aerobically. And the reasons for that are two things that I'll just summarize again. One is you don't make much ATP. Compared to the 30-something ATP that aerobic respiration produces per glucose, this is small change. But you can go through large amounts of it, and so it can make a lot of ATP. It's just in a very inefficient way of going about it. The second thing is the acidity problem that we talked about. If you're producing a lot of acid, muscles can't keep doing that forever, which is why fatigue sets in fairly quickly. Okay. Last thing. So myoglobin was basically, oxygen backup for aerobic respiration. Creatine phosphate is basically phosphate backup in a way it's not actually anaerobic respiration, but it's similar. And the idea is creatine phosphate, which they're abbreviating CP, can take its phosphate, hand it off to ADP to make ATP. This isn't anaerobic respiration, it's not aerobic respiration, it's just phosphate storage. Creatine is holding onto that phosphate, and when muscles contract intensely and you're in a pinch, it'll hand off that phosphate to ADP, and you wind up with ATP. The other end product then is just plain old creatine, right? No phosphate. They really should just make this a big C because the phosphate is gone. This is what's happening during intense muscle contraction. Later on, when you're resting and you have extra ATP to spare, I should write that down here, you can have ATP give its phosphate back to creatine to remake creatine phosphate, which leaves you with just plain old ADP again. It may seem kind of strange that you use creatine phosphate to make ATP, and then later on you use ATP just to remake creatine phosphate. The difference is one happens during contraction and one happens during rest. And so all it's saying is you keep these extra phosphates around for when you're in a pinch and need extra ATP production, and then when you're resting and you have extra ATP, just remake the backup again. It's fairly straightforward. Creatine kinase is just the enzyme that makes that reaction happen. Okay, so to pull it all together, this is a graph from a data set in a paper where they asked volunteers, which I hope got paid a lot of money, to sprint really intensely on a bike. And at certain time intervals, the researcher took a muscle biopsy, which basically means they stabbed a giant needle into their quadricep and took a chunk of muscle tissue out, and then did a chemical analysis to see how much ATP they were making and where exactly that ATP was coming from. So the summary is time zero is when they started their sprint, and y-axis is ATP turnover. If you look at the key up here, what it's showing you is the white bars are creatine, the slashed bars are anaerobic respiration, and the black bars are aerobic respiration. And the thing that I really want to point out is it's tempting to think that these are sequential, meaning a lot of people have the idea that muscles start out aerobically and then become anaerobic only when you run out of oxygen. But in reality, when you start off doing something very quickly, right, these people were asked to sprint as hard as they possibly could on the bike right from the start, aerobic respiration just can't possibly produce that much ATP that quickly. And so in that first 6-second burst, they're relying almost entirely on creatine and anaerobic respiration. And over time, those bars drop dramatically and the aerobic part increases. The aerobic increase is because the person's respiratory rate and heart rate are going up and they can just deliver more oxygen. Creatine levels are going down, obviously, because they're just running out of creatine phosphate. They don't have any more left. Anaerobic respiration is going down because they're becoming acidic, and muscles can't be acidic for very long. And that's sort of the feeling of fatigue that a person will get. They start losing the ability to use those glycolytic enzymes because acid levels are increasing. Of course, if you let them rest for a very long time, they'll eventually go back to being close to full performance. But that's just the time that it takes to flush out all of those metabolic wastes and acid that built up. All right. So to wrap all this up and tie it together, let's look at muscle fiber types. And all this is saying is that within one muscle, there are different muscle fibers that specialize in different kinds of ATP production. And they're called either oxidative or glycolytic. Oxidative means aerobic, and glycolytic means anaerobic. And slow or fast depending on the speed at which they produce ATP. Type 1 fibers are aerobic. They mostly use oxygen, which is why they have lots of myoglobin. They don't have much glycogen because they pull their nutrients almost exclusively from the blood, which works because that has to be done fairly slowly. And they're recruited first because they're efficient, right? You'll always choose to use the most efficient means first, and these type 1 fibers are slow, but when you're doing something slow and easy, that's perfectly fine, which means that they're resistant to fatigue. And the reason they can do this is they're packed full of plenty of mitochondria and blood vessels to deliver that oxygen, and mitochondria to actually use that oxygen to produce ATP efficiently. If the contraction intensity goes up and you need to recruit bigger, stronger muscle fibers, eventually you'll tap into these 2As, the fast oxidative fibers, which are still primarily aerobic, but they can do some anaerobic respiration. They produce ATP at a faster rate, but because of that, they fatigue a little bit more quickly, and so they can't be used for an incredibly long time. If you really dramatically increase the contraction intensity, you'll have to start tapping into those glycolytic fibers, which are anaerobic, which explains why they don't have much myoglobin, but they have lots of glycogen, because it's impossible to pull nutrients out of the blood that fast, and so they have to use internally stored glycogen to fuel that anaerobic glycolysis. Remember, glycogen is just stored polymerized glucose that can be broken down and used internally inside the muscle. Because that anaerobic glycolysis produces so much acid, muscles fatigue quickly, because again, they can't acid burn themselves from the inside out. And they also don't have that same capillary and mitochondrial density that type 1s do, because, well, they're not aerobic anyway. There's no need for oxygen delivering and oxygen consuming capacities, right? And so for the exam, mostly focus on comparing the extremes here, type 1s and type 2Bs. Think about why the recruitment order goes in the way it does, based on efficiency and based on ability. All right, I think that's where I'm going to stop. The important stuff on the next half dozen pages is review, mostly from exams 2 and 3. And so you can go back to those videos and find recaps of motor cortex information and reflex pathways that we talked about before.